Next up in proteins is part 3, from gene to protein to function. So in this we're going to talk about where these protein sequences come from, remembering that the protein sequence is the primary structure. That should not be news to anyone. We're going to identify the central dogma, justify why proteins must be purified for study, and provide examples of post-translational modifications. So 2 and 3 kind of really go together quite well. Okay, first off, where do proteins come from, right? They come from the central dogma. Ha ha ha. So we have genes in the DNA, regions that code for specific mRNAs. Remember, the DNA encodes for mRNA, tRNA, and rRNA, but in terms of the central dogma, we ignore those contributions because those don't directly use the sequence to make the final protein. Okay. DNA can also make itself through the process of DNA replication. DNA to mRNA is the process of transcription because we're using the nucleic acid language to the next nucleic acid language. When we translate, we're changing the language from nucleic acid to protein. So remember this process. The DNA be like TAC this is written in codons. Codons are a series of 3 nucleic acids, then become RNA, which doesn't use T anymore, it uses U. So this would be series of 3, this is another codon. And then the mRNA codons you can use for the codon table to determine what amino acid would go there. So this would be methionine or M. Okay. So the protein's primary structure is based on the DNA. And remember, we have a lot of DNA information because of all of these genome projects that have been going on since the late 1990s and early 2000s. We have the human genome, influenza. These are constantly being dumped into databases that everyone has access to. So it's very, very easy to get the protein primary structure information. However, the protein primary structure information doesn't tell us what the gene information doesn't tell us what the final tertiary or quaternary structure of the protein would be. And that's because of post-translational modification. So here's the process of translation. The ribosome will spit out a protein. So here's our ribosome, right? And it's over the mRNA, and out of the mRNA, the tRNAs go in there, right? They look like T's, and they have the amino acids. So the amino acids will be a growing chain coming out of the ribosome. However, they haven't folded yet. So folding is one post-translational modification. This is done by a special class of proteins called chaperones, whose job it is to bind the growing peptide chain and put it into its correct 3D form, the tertiary structure. Additionally, the gene information only gives us one protein chain. So this would only tell us the chain of one protein. What if in the actual solution we have 2 proteins working together to form quaternary structure? The gene information wouldn't tell us that. Other post-translational modifications include if cleavage is necessary for the protein to function, such as what happens in proinsulin, which is a protein hormone. This becomes insulin in the body only when it's needed. And that's done by cutting part of the protein out to make it active. Also, because methionine is the automatic start codon for many, many, many proteins, sometimes it's not necessary for function. It was just the signal, like the green light to go. So occasionally methionine can be removed. if it's not necessary for the structure. And also, any covalent modifications. Maybe there's a cofactor that's needed that needs to be covalently added to the active site. None of these are encoded in the DNA information. And so for that reason, it's very, very important to take these proteins that are in solution and be able to purify them away from all the other stuff, like the salts, the other proteins, the DNA and the RNA, all of that, in order to get just the proteins that we were looking at. And so that's done through protein purification. It removes all the contaminants so we can figure out the protein's structural information just by itself without any confusing information in there. Okay, and so how do we purify a protein? We use the protein's unique characteristics, and typically each protein has its own unique PI, and it's got its own unique UV extinction coefficient and its own unique activity. So we can run this through a series of columns. So we do column chromatography based on salts. Column chromatography based on salts. Sometimes we can tag a protein with something that we can then, and it's called an affinity tag, to pull it down. So for example, histidine has high positive charge. So if we have if we add a bunch of histidines to the C-terminal or N-terminal end of a protein, then we can use a nickel column in order to pull the histidines out, because histidine is very attracted to nickel. So we use column chromatography based on the structural information of the protein in order to pull it out of solution and remove all of those other things. We can also use electrophoresis to separate proteins based on size and based on charge, because every protein has its own combination of amino acids which have different charges. So we're able to separate proteins based on size and charge that way.